Hello friends, welcome to Simplified Biology. Today's topic is development of embryo sac in angiosperms. Angiosperms are the flowering plants. So the reproductive part of angiosperms are the flowers. And the female reproductive part is gynoecium, which is made up of pistil or carpel. The pistil or carpel consists of ovary, style and stigma. The embryo sac is the female gametophyte which develops from the megaspore. Now developing inside the ovary are the ovules. Ovules are the megasporangium, and inside the ovule develop the megaspores, from which develop the embryo sac or the female gametophyte. An ovule or the megasporangium has a central mass of cells called the nucellus, surrounded by two integuments, except at the region called the micropyle. Or you can say a small opening is present called the micropyle. Stalk of the ovule is known as the funicle. There are some plants in which the ovule has a single integument, that is known as unitegmic, seen in case of monocots and in members of family Fabaceae. Fabaceae is your pea family. Ovule having two integuments are referred as bitegmic, seen in members of family Asteraceae, that is your sunflower family, while some ovules are without any integuments. They are referred as ategmic, example of which is Centellum. In some ovules, a third integument in the form of an outgrowth is found present. That is referred as an aril. Best example of which is lychee. Aril is an edible part of lychee. Now development of embryo sac. During development of ovule, a hypodermal cell towards the micropyle differentiates. A hypodermal cell towards the micropyle differentiates. It behaves as the archisporial cell. Now the archisporial cell either directly behaves as the sporogenous cell, or the archisporial cell divides to produce two cells, of which the outer is the parietal cell that forms the wall layer, while the inner is the sporogenous cell. This sporogenous cell behaves as the megaspore mother cell. The megaspore mother cell undergoes meiosis to form 4 megaspores. Formation of megaspores from megaspore mother cell due to meiosis is known as megasporogenesis. Now, of the 4 megaspores that are formed, 3 that are present towards the micropylar end degenerate, while the 4th on the opposite side is the functional megaspore. The functional megaspore increases in size to form the embryo sac, while its nucleus divides twice, forming 8 nuclei. These 8 nuclei arrange themselves in the embryo sac. 3 nuclei arrange towards the micropylar end. They form the egg apparatus, which has a central egg and 2 synergids. These are the 2 synergids. While 3 arrange themselves towards the opposite side, they form the antipodal cells. Antipodal cells are the only vegetative cells of the female gametophyte. While 2 nuclei, they lie at the center, they form the upper and the lower polar nuclei. While the embryo sac cell is referred as the central cell. Since only one megaspore takes part in the formation of the embryo sac, it is referred as the monosporic type. When 2 megaspore take part, it is bisporic, and when all 4 take part, it is tetrasporic. But monosporic is the most common type. So the most common type of embryo sac is monosporic, 7-celled, 8-nucleated type. Seven-celled, first cell is the central cell, then the 3 antipodal cells, an egg and the 2 synergids. The egg and synergids together form the egg apparatus, which lie towards the micropylar end, while the 3 antipodal cells lie towards the opposite side, that is towards the chalaza end. The end opposite to the micropyle is known as chalaza, while the 2 polar nuclei, they lie at the center. So this is how embryo development takes place in the angiosperm. Thank you for watching.